Hi, everyone. We'll get started and shortly. We're just letting folks join. We still have quite a few people joining, so thank you for your patience. We'll get started in just a minute or two. We still have a steady stream, which is very exciting. Okay, all right. Well, I think we're going to go ahead and get started. I am super excited today. So for those, my name is Claudia Regal. I'm the director of the City of New Orleans Mosquito, Termite, and Rodent Control Board. And also heading up the Gulf South Vector Educational Center for Training, Outreach, and Resources. I know some of you out there have been with us already with our two previous webinars. And today is going to be the final installment of this particular series. And we're going to be talking, Mike Riles is going to be talking about biology and ecology of invasive mosquitoes in the southeastern part of the U.S. Now, I wanted to give everybody a few little reminders before I introduce Mike. If you could kindly leave your stay on mute, please, and use the chat in order to ask us some questions. I've got Miss Astacia is in the background with us, and she's monitoring the chat. So we'll make sure that we bring all your questions to Mike. And we've had lots of questions about this. We're not sharing the presentation, but what we are doing is we will take this recording of of all three sessions, and we're going to put them online at the City of New Orleans Mosquito Control Board, and then also we'll make it available through the Gulf South Vector. It's going to take us a little bit of time because there's some new requirements and we have to make sure that the videos are, compliant, but it is going to get there. The one thing that you might consider so that you stay in touch what's coming new from Gulf South Vector is to sign up at www.vectoroutreach.org. And we're going to put that in the chat for you. And if you go to the bottom of the landing page, there's a subscriber section. And if you subscribe, we're going to be sending you some notifications of some cool things that are coming up. At the very end of this presentation, we'll talk a little bit more about another series that we have, which is all about emergency response. So I want to take just a minute and introduce our speaker today. His name is Mike Riles. He's a vector biologist and entomologist with over 15 years of experience in mosquito ecology, surveillance, and integrated pest management. He is the regional sales manager for Central Life Sciences, supporting mosquito control programs across the Southeastern United States with technical guidance and field-based IPM strategies. He previously, for about 9 years, was an entomologist at Beach Mosquito Control. So for the folks that are out of state, that's in Florida. And now he co-directs the multi-state Mosquito Beacons initiative focused on invasive species and taxonomy. So that's what you're going to hear about today. He's co-authored 18 peer-reviewed publications, and super exciting, he is finishing up a master's in entomology from the University of Nebraska-Lincoln. So for those that don't know Mike, he's an amazing taxonomist. And so we're so excited to have you here today, Mike, for the next 2 hours. So I'm going to kick it over to you, and everyone enjoy. And don't forget to put your questions in the chat.
Thank you, Claudia. I appreciate that introduction, and I also appreciate the invitation as a co-director of Beacons to come in and support Gulf South Vector and their the goal of disseminating information to vector groups across the southern U.S. As Claudia was saying, my name is Mike Riles, and I'm going to be your technician for the next couple of hours. Hopefully we keep things kind of in time, but I may go over a little bit, so hopefully I don't ruin too much of your Friday before lunch. So I just want to jump right in and talk about beacons for a minute, and then we're going to talk about what the definition of an invasive species is versus a native species versus a non-native species. I'm going to talk about identification books, kind of like this one I've got propped up here, to kind of drive home the message of what's important when we're getting ready to do identifications. Then we're going to jump into the biology and ecology of the mosquitoes in the Southeast U.S. that we feel are non-native or invasive. Y'all ready? Right, here we go. So, introduce beacons. Beacons is the biodiversity enhancement and control of non-native species. This was the driving force behind the idea of beacons, and our motto is the shining light on invasive and non-native mosquito species of the South. The overall goal of our working group is to increase the capacity for non-native invasive mosquito surveillance and control across the South, while also raising awareness of new and potential introductions to help frontline mosquito control workers identify new threats. So that's basically our goal throughout this whole process of being in business for the last 6 years. We are a multi-state working group, as Dr. Rigel was saying. We are mosquito control professionals, private pest control professionals, public health professionals, and then research and extension partners within academia. This is our current board of directors. And these are our current members that we call stakeholders across the entire southern U.S. So this encompasses everything from Virginia to Texas, all the way to Puerto Rico in the Caribbean. We are ranging with municipal groups that are local, state groups that define those other groups, and then we have some federal stakeholders as with the CDC. So let's jump into definition. So before we jump into the identification, biology, ecology of these really cool insect pests, we need to define what an invasive species is. So this has been ongoing since 1999. An executive order was signed by the late President Clinton. The the order required a council of departments, and so what was formed was the National Invasive Species Council. This was established then. As per that executive order, invasive species is a species that, one, is non-native to the ecosystem under consideration, and two, whose introduction causes or is likely to cause economic or environmental or harm to human health. Native species are organisms that are part of the balance of nature that has developed over hundreds or thousands of years in a particular region or ecosystem. So there's this evolutionary process of adaptability over time in habitats that define what a native species is. A non-native species, those are organisms living in areas where they do not naturally exist. An invasive species are non-native species, right, to the ecosystem under the consideration that we're kind of viewing through our observations, whose introduction causes or is likely to cause economic or environmental or harm to human health, as we were describing previously on the last slide. Non-native species and invasive species cannot be used interchangeably. This was a lesson I learned about 6 years ago. We like to make sure that those designations are not intertwined. They are definitely separate. So let's look at introductions. Mosquitoes can be transported across all 4 life stages as the adult and as the 3 forms of the immature stage. So with adults, it can be man-made constructs, vehicles, things that we make and causing movement from one place to the next, whether it was 300 years ago in a water barrel, on a ship crossing the Atlantic, or possibly even a plane or a cargo ship today, coming from Southeast Asia into the Western Hemisphere. So when we're thinking about those types of things, we're going to be talking about the tire trade, plant trade, transport of containers that can hold water, floods, and then we can think about all the different life stages that can be incorporated into those types of containers across the board. And this is going to be something we're going to be seeing for the rest of the day. It's going to be a common trend as we move forward through the slide deck. So let's talk about barriers of those introductions. You know, if there's an invasion of an organism into an area, what kind of barriers are going to hinder that from actually grabbing hold of that ecosystem and becoming habitable into that habitat? So we have geographic barriers, we have survival barriers, and then we have dispersal and spread barriers. So there's quite a few things that are going on that it can actually hinder the invasion. And so when we do have an invasive make it through all of this stuff, it's pretty impressive of the robustness and the tenacity of that organism to go through all the things that it's going through to be able to establish itself. So with geographic barriers, we're looking at the presence, absence of the preferred habitat. We can also talk about the adaptability to habitat that might not be so preferred. We're talking about landscape features that promote or obstruct the movement of the organism from one place to the next. And let's talk about survival. So we have competition, predation, host range. We have this whole intricate food web that's already going on, and that organism comes in and interjects within these feedback loops that are going on for resources. So there's a lot of complexity going on from the ecological perspective, and then how it's moving, and then how it spreads. So we kind of talked about the anthropogenic type movement that we cause and give the capacity to create taxis for these organisms, right? So, but we're also looking at the actual motility of the organism itself, and also its behavior on feeding. That direction of range of hosts, is it going to be palatable to the organism? And then is its behavior going to be able to meet the parameters of what those hosts are giving as a resource within that food web interstructure in the ecosystem? So let's talk about the biology. So there's common traits of invasive non-native mosquito species. We're going to see these trends as we move through these pests that we're going to be talking about today, where you have desiccant resistant eggs, and then you have delayed hatch type mechanisms that prolong the propagation of the pest over time. And then we want to talk about oviposition preference. Where is the pest actually ovipositing her eggs? Does she have an affinity for natural containers or man-made containers? Hell, possibly even not even containers. Um, some of the mosquitoes we're going to be talking about today are ovipositing in muck and in egg rafts and grassy furrows and things like that. And then we go back to that anthropogenic type instance where we have these human dominated landscapes where we're changing the landscape and we're occupying these spaces, not only to live and do commerce, but also to grow the food that we need to sustain our growing populations. All of these instances are creating all of these new habitats that these invasives actually find rather cozy. So expanding human population generates those new microhabitats where they can thrive. And this is interesting within the food web. There are no natural predators for these invasives as they've moved in. They can encroach, establish, and propagate without the realization of predators. Well, except for us, right? Because we're mosquito control and we like to kill the invasive pests, right? So if we're doing our job well, we actually are the natural predator for invasives. So, um, hold on, I'm having an issue with my slides. Here we go. So when we think about invasion, we want to think about things in a curve. And this is an interesting infographic that kind of gives you a realistic view on what control is. So that was a real nice segue about us being the predator. And here is us predating against the pest, right? So as time goes on, invasive species are more costly to manage. If we have an indicator of an invasive and we do some prevention and eradicate it, in the long run, if we're putting some serious resource to that eradication, we are seriously saving us time, resources, and money in the long run. Because once an invasive species becomes established, it is rarely possible to eradicate. And one of the only things that almost eradicated Aedes aegypti was the satirization impacts ecologically with Aedes albopictus. She was actually our friend for quite some time, but there's this other roundabout way and this reemergence pattern that's happening because of the adaptability of these things. So when we think about when there is an invasive and it's something we really need to target, we need to think about 10 years down the road, how much that's going to cost us resource wise in the long run. So we just need to put our resources in the beginning to knock that pest out. Excuse me. This slide is basically an overview of the invasive and non-native mosquito species introductions into the southern United States from 1920 all the way to 2023. From Culex coronators introduction and slow spread throughout the United States, all the way to uh, Aedes japonicus, Aedes albopictus. Aedes japonicus moved through temperate regions into subtropical zones. Introductions like Culex lactator, Aedes pertinax that aren't container inhabiting mosquitoes. How did they get here? We're going to be chatting about that throughout the day. And then we have like Aedes vittatus, which has been found at the naval station there in Guantanamo, Cuba, coming to a container near you in the peninsular area of Florida. Okay, so I'm going to give about 30 seconds for everybody to grab this barcode on this QR code here on this slide. This is a online invasive mosquito dashboard that Dr. Lindsay Campbell and her team have designed within the Beacons group. It's got a really nice platform of invasive and non-native mosquito established locations throughout the southeast United States. This is a great resource, not only for a reference, but you actually add your information to this dashboard through communicating with the Beacons group. All right, 10 more seconds. If you're having trouble with that, just contact us and we'll get you squared away. All right, as you're seeing here, this is the identification geographical distribution of the mosquitoes of North America north of Mexico, third edition that Dr. Nathan Burkett Cadena revised for us. It was a much needed revision for North America and Canada. You have to use a key when you have a mosquito you're not able to identify. Most insect keys begin with genus. Once you're sure of the genre, you move on to the species key for that next genus. If you've attended the last two courses within this webinar series, you are well familiar about dichotomous keys and the things you have at your desk to make references. Within the beacons group, we talk about multiple different identification keys at your desk. If you can see back here in my bookshelf, the second shelf is all my mosquito identification keys. I've got about 12 different volumes of reference that I use when I'm at my desk. I'm doing identification just in case. We're talking about continental keys. We're talking about global keys, regional keys, state keys, county keys, district keys. All the way across the board, we really recommend that you use multiple keys at your desk to make your determinations, especially if mosquitoes are not in the keys that you have readily available. So this is just an infographic that shows how the keys we use here in this area and their overlap. So it's really interesting when you have overlapping keys. The information sometimes in those keys per species can be different. So there's always a different take taxonomically from one author to the next. There might be something in those keys that can help you push forward in your identifications. All right. Thank you for that journey and the introduction into the biology and ecology of the non-native and invasive mosquitoes in the southeastern United States. This slide is kind of depicting the range of scope of material that we're going to be discussing throughout the next few slides. Well, actually, probably 50 slides. We look at the bionomics. We look at the habitats, the distribution and range, their dispersal strategies, collection methods, host preference, and their transmission status for pathogenic transmission, along with the morphology. So the way that we're going to orient this, there's going to be a slide, and then we're going to talk about the morphology. And in some instances, I'm going to be grouping mosquitoes together for similarities and differences. I will be moving quite, not quickly, but not hurriedly, but we will be moving through these at a decent pace. So I hope that you brought your dancing shoes. First mosquito we're going to talk about is Aedes squamipennis. Aedes squamipennis is a new introduction into the United States. Her native range is South America into Central America. Her non-native range currently is in the southern tip of the peninsular area of Florida. She is associated with aquatic vegetation in the habitats. One other thing that's going on with this mosquito is interesting. Not only is she attempting to enter the North American continent through the peninsular area of Florida, there are current reports and publications within Mexico that she is coming up the eastern coast of Mexico, and the last record was about, I think, 300 miles from the southern border of Texas. So she now is encroaching up onto North America from two different approaches, which is pretty interesting to watch. Now, her larval habitat is interesting, where wetlands that support dense growth of water lettuce. I've been pulling her in South Florida out of, she is adapted to duckweed. In sunlit type areas, you can pull Aedes squamipennis larvae out of the habitat. Now, her dispersal strategies, her flight range in kilometers around 9.6 kilometers. How did she get here? That's kind of curious. And so everyone put their thinking caps on. As we move through these slides, we're going to come into these instances where we have mosquitoes that show up here, but they're not container inhabiting mosquitoes. She does collect well in larval collections with dippers and things of that nature. CDC light traps baited with carbon dioxide as the attractant seem to work well in southern Florida. Currently, we know that her host preference are birds, and there is some pathogen transmission going on in her native range for this mosquito. Now let's get to the cool part. Now this is what this mosquito looks like. She looks way different from most mosquitoes in North America, with some decent similarities to like a Colquittida perturbans or a Mansonia titilans or a Mansoniidae, with the peppered look on the wings. So let's look through some of this morphology as we move forward. She is a large, peppered, multicolored scales with banded legs. So these scales are goldish yellow looking with bright white ivory looking scales, and then dark, almost dark brown scales throughout the entire mosquito. Let's zoom in on 1A, where the enlarged view of the head, scutum, palps, and thorax really give you a nice view of the diversity of patterns of scales throughout, and then the coloration differences over the entire body of this mosquito. So number 2, this is where things get really interesting, where the yellowish scales covering the clypeus, the torus, and the flagellum 1, it's very ornate. So when we're looking at flagellum 1, you're seeing that those yellow scales on the interior of the flagellum 1. You're seeing these small little white patches of ivory scales on the clypeus. And then when we looked at the tip of the palps, which are banded and dark banded with golden yellow scales, banded black with a wide band, and then tipped with those ivory scales, it pops. When we're looking at the wing, it's very ornate. And this is where this mosquito kind of looks like other plant-associated mosquitoes like Mansonia colquittida, but the pattern are similar, but the colors are wide-ranging, diverse. Broad, light, dark scales on the veins with direct, dark white scale tufts along the fringe. Number 4, this is the real interesting trait character that we're looking at on the apex of the mid and high femur. There are these erect scale tufts that are very pronounced. You cannot not see this. It's a pretty amazing character. I actually wish that we had a mosquito in front of you right now, because everybody would be going, oh, wow, this mosquito looks amazing, versus what we normally see in our scopes here in North America. All right, now we're going to jump into some familiar ground. And I know some of this information is Common Core, so the next couple of mosquitoes, we're just going to kind of tear through this, and we're going to jump into some complex identification processes with Aedes aegypti, Aedes albopictus, Aedes nodoscriptus, all right? So Common Core, aegypti has been here for quite some time. She's considered a naturalized invasive mosquito. Her invasive range in the United States is all the way across the southern United States, up the eastern seaboard. Her collection is basically, for larvae, you're using dippers and basters, BG sentinel traps, BG lure traps, sometimes low-hanging Fay Prince traps. She does associate with colors like dark and white for attraction. She has a very short flight range, desiccant tolerant eggs, and this is going to be a common core that we're going to talk about the next few mosquitoes. And then the host biting preference. It's us, right? I mean, she's so adapted to us and what we're doing, and this makes her a really good vector for a wide range of different pathogens, from yellow fever, dengue, chikungunya, Zika, and she's a potential vector of a couple different encephalitic viruses. So let's just go over the morphology. As we all know, with Aedes aegypti, she has that very popping lyre-shaped patch of white scales on her scutum. She moved to number 2, and this is very important. The white scales on the clypeus. On number 3, this is important as well. There's a solid white abdominal sternus all the way up underneath. Aedes albopictus does not have that. Number 4 is another great character that you can pull this mosquito from her Stegomyia sister, the white band on the dorsal middle femur. It's there on her, but not albopictus. We're not really going to talk about larval comb scales. We are going to move on to Aedes albopictus. So Aedes albopictus is a similar story, but later on in time, where this mosquito adapted to us and tires and containers, and has moved like wildfire across the entire globe. Her invasive range is very impressive, and the speed that she conducted this range is seriously impressive. She can outcompete other container mosquitoes. She's an opportunistic ovipositor and will oviposit in a variety of artificial and natural containers all the way across the board. And she is associated with us in our home, but she's also associated with those sylvatic fringe environments that we see in between residences, commercial properties, and things of that nature. Egg desiccation, just like Aedes aegypti. This mosquito is very intent on grabbing a blood meal, and she'll follow you just like Aedes aegypti will. Collection methods all the way across the board, super similar to Aedes aegypti. Same collection methods. Host biting preference is a little bit different. So her status as a vector in the United States is considered secondary to primary versus Aedes aegypti. She does like that kind of crepuscular times to feed. So she is an opportunistic feeder and will occasionally bite birds, large mammals, us, domestic pets. In some instances, blood meals have found, some herps as well. So she has a wide range of capacity for the pathogen transmission across the board. She does have the same similar platform as Aedes aegypti, and she is the potential vector of things like West Nile virus and sometimes considered a tertiary possible contributor to La Crosse virus transmission cycles. So, Aedes albopictus. This is a very interesting looking mosquito, and there are some similarities across the board, and we'll talk about those. One of the distinct differences with this mosquito is the white medial stripe down the dorsal of the scutum. Let's take a look at that clypeus. We were looking at clypeus on Aedes aegypti. There were scales present. On Aedes albopictus, the scales are not present. When we're looking at the abdominal sternus, they are white banded on Aedes albopictus. There is no white band on the middle dorsal femur of Aedes albopictus. All right, who thought we were going to be talking about a mosquito from Australia, right? Well, Aedes nodoscriptus, as native's range is Australia, Tasmania. Her invasive range is some incursions in Southeast Asia and 3 counties in Southern California. Cryptic in morphology, regionally suggesting a diverse phenotypic morphologic complex where coloration of scales can be different. She has a wide range of artificial and natural containers that she'll abide in her immature forms. Everything from bamboo stumps, leaf axils, rock pools, palm fronds, different types of man-made containers across the board. She is a container breeder that uses diapause eggs, and she does have desiccant-resistant eggs. She's spread by humans, just like Aedes albopictus and Aedes aegypti. So there's some similarity trends as we're looking. Collection methods are similar to the last 2 mosquitoes. She will come to a wide range of different types of container inhabiting mosquito traps, but she'll also come to gravid traps, baited with some sort of fermented juice that you can put into those traps as an attractant. Now, she does have a very wide range of host preference to us, our pets. She will feed on birds. In Australia, she's fed on marsupials. She will also feed on bats. She has a wide range of pathogens that she can um, pass on, from Barma forest virus, chikungunya, dengue, Ross River virus, Zika, and then dog heartworm. So this is a small to medium-sized species. Are we seeing something similar with this mosquito versus Aedes aegypti and Aedes albopictus? So let's just jump to 4 real fast. There's a lyre-shaped lateral silvery scales all the way across. But when you look at number 4, look at those 2 lateral medial lines going down that medial white stripe. What are you seeing different there? So let's kick back real quick to Aedes aegypti. When we're looking at number 1 and we're looking at those lines going down, they're not very centered. There's like these lateral medial lines. When we look at Aedes nodoscriptus, you see a center medial line, kind of like Aedes albopictus, but it's more pinstripe. What you do see on the lateral sides of either that pinstripe are 2 golden scaled stripes on the scutum. This is very pronounced. Let's jump to 3. Its proboscis is banded. The wings are scaled dorsally throughout, similar to the other 2. But when we jump to number 6, things get a little bit different. The pedicel is not scaled exteriorly, only interiorly. This is a secondary character that you're not going to see in keys. The palps are tipped with silvery scales, and guess what? The clypeus is bare. So when we were looking at albopictus, her clypeus was bare. But when we're looking at Aedes aegypti, her clypeus has scales. So when we're looking at these mosquitoes, we're going to start noticing some differences. So let's group these mosquitoes together. This is something I challenge my in-face classes with. I have each of these mosquitoes in front of them. In some instances, I'll throw an Aedes midioviditatus in there just to kind of shake it up a little bit. But when we're looking at these mosquitoes from a lateral view, what do you think? They look seriously similar. And you can make comments in the chat box. Do you think these mosquitoes look similar, even though I've got them named as different? Look at the banded legs. Look at the abdomens. Look at the wings. Look at the overall thoracic scaling patterns from this far away on these pinpoints. There's some serious similarities across, and at gross levels, you could totally say that all these mosquitoes were the same. But let's take a closer look. When we're looking at Aedes aegypti, and we're looking at Aedes nodoscriptus, and we're looking at those lyre-like markings, you can see similar scutal around the scutal fossa area where those curvatures are coming in and you're lining down. But then what's different are those medial stripes going down the center or off center of the scutum. We can also take a look at the scutellum. They are arranged with setae and scalings, but in some instances, these scalings on the scutellum are different. And at the bottom end of the scutum, you have some different style lines going on. They could be wide, they could be separate, they could not be touching. So these are things that I challenge you to look at when you're viewing mosquitoes under your scope. What are the differences between mosquitoes that you might be seeing in your scope? Now, I'm really going to challenge you. Let's look at the clypeus, the pedicel, and the proboscis. These are all secondary, tertiary character states. Some of these are in keys. Some of them are used as internal measures to separate like an Aedes nodoscriptus from an Aedes aegypti. So when we're looking at these 3 structures, I want you to take note of the presence, absence of patterns. The pedicel on Aedes aegypti is scaled exteriorly and interiorly. When we're looking at that same structure where the flagellomere are rising out of, the Aedes albopictus and Aedes nodoscriptus are scaled interiorly. Let's look at the palps on Aedes aegypti, Aedes albopictus, and Aedes nondescriptus. The palps are tipped with silvery scales, can be variable. When you're looking at albopictus, they can be variable. Palps tipped with silvery scales can be variable among nondescriptus. So is that a character that we can use? Think about it. Clypeus on Aedes aegypti is scaled, but not scaled on either Aedes albopictus or Aedes nondescriptus. This is something that you can pull apart, especially when we're thinking about scutums being totally jacked up from fan manipulation, and all those scutums can be rubbed off. So with Aedes albopictus and Aedes aegypti, there's some differences from Aedes nondescriptus with the proboscis. It's totally banded on Aedes nondescriptus. So when we're viewing these three mosquitoes that are fairly common in containers in the areas that they actually are established in some instances, these mosquitoes are cohabitating, sometimes in the same container, sometimes the same trap. So these types of character states can really assist you with moving through the piles of mosquitoes that all look similar in your trap net. All right, here's another container inhabiting mosquito, Aedes vittatus. Not currently in North America, but now has dipped into the Yucatan, and there is some urbanization spread in Mexico. There's some preprints out about its encroachment in urban areas in Mexico. It's in Cuba, Dominican Republic, Jamaica. There are reports of it being in Guantanamo Bay, which is U.S. property. So maybe we could say its larval habitat's a little bit different than some of the other container inhabiting mosquitoes that we've been looking at. Rock pools, tree holes, serious depressions that can be made from large mammals, domestic containers. It is egg desiccate tolerant. So we're seeing a pattern here with the 4 mosquitoes. Caribbean introductions are speculated to have occurred via shipping container transports, just like some of these other mosquitoes we're talking about. We're starting to see some common trends. So its native range is Asia, Southern Asia, throughout Africa, a little bit of Southeast Asia, and it's being carried here in containers. Like other invasive Aedes, you're not really going to collect this mosquito collecting using CDC traps, unless maybe you add a little bit of spray paint, black, put it down about 18 inches off the ground. We really suggest to use BG type sentinel traps with lures. The surveying potential production sites, artificial man-made containers is necessary. More surveillance needs to occur, because now it's extended its invasive range into these habitats here in the Caribbean and to Mexico. She does have a wide range of preference for host biting. She's an opportunistic. She will feed on primates, domestic animals, rodents, us, bats, birds. So her pathogen transmission platform is quite extensive. I'm not going to go through all of these. There's I think there's like 10 or 11 here, but look at some of the similarities we will make note of, like the Zika virus, yellow fever virus, chikungunya virus, dengue virus. Guess what? We have all those in the Western Hemisphere. So this is of concern. This is one of the coolest mosquitoes. Very ornate, very popping with its silvery white scales. And when we're thinking about silvery white scales, ivory type scales, when we're looking at it in number 1 from this lateral view, you're seeing these very broad bands on its hind legs. We're also seeing ornate patterns on the abdomen area, the thoracic area beneath the scutum has a wide range of different type of scaling patterns throughout the entire area. Let's move to number 2. So instead of a stripe, she has dots, these little spots that are to the medial line, lateral, going down the scutum. Look at number 2, 2A. the one in the top is an actual specimen from the Dominican Republic. Number two is a specimen that we got some photographs from David Pecor up at the Smithsonian. But you can see these lateral type spots going down. They pop out. But one thing I want you to take note of is number three. That tibia, that hind tibia, there's a subbasal white spot and a white band, approximately a third of the way from the tarsi. So when you're looking at that femur and think about Psorophora species in the United States, Psorophora columbiae has that character. When we're looking at number four and we're really diving in, we're looking at the meso postnotum and the presporacular area are bare. It's interesting, even with all the ornate scaling that's going on. Guess what? Let's take a look at number five, which is below number four. For some reason, the number is not there. The proboscis is darkly scaled with sporadic pale scales. Interesting orientation. The clypeus with bilateral small patches of narrow pale scales. And these things are like just little dots, bink, 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 throughout the clypeus in a random pattern. The palps have pale scales at the tip and a band halfway. The pedicel with exterior and interior pale scales. So let's backtrack. Let's remember, which other container mosquito do we have in North America that has the pedicel with exterior and interior pale scales? I knew you'd get it right. Aedes aegypti, right? So there are some similarities with these things that we're going to find in containers. Currently, this mosquito is in and around environments that she's associating with Aedes aegypti in the Caribbean and in Mexico. So let's move to number six, where the abdomen has large median white spots. The basal bands have lateral curved white markings. These are disparate from the bands, detached with very ornate patterns laterally coming down the abdomen, giving it a fan-like look as you move down the abdomen. I'm going to take 2 seconds. I need a break. All right, I appreciate that break. All right, now we're going to move to another container inhabiting mosquito. This mosquito is Aedes japonicus. Its native range is basically Japan and the Korean Peninsula. She came here just like Aedes albopictus, moving like Aedes vittatus, cruising across the Atlantic or the Pacific, just like Aedes nondescriptus. Her expansion is slower, and that has to do with her ecological adaptability within temperate climates. She was introduced into North America and Europe in the mid-late 1990s. She cascaded throughout North America from 2014 to 2024, from Florida all the way to Nebraska. She's currently adapting to subtropical climates, as within Hawaii and the panhandle of Florida. We do need more collection records for this mosquito. I think Dr. Bill Sams, the famous Army medical entomologist from Texas, is kind of working on that paper currently. So her native habitat is rock pools in her native range. But guess what? Her larval habitat in North America is also rock pools. She's totally displaced Aedes atropalpus, the eastern treehole or the eastern rock pool mosquito, in riverine areas throughout the eastern seaboard. She will also oviposit in natural and artificial containers across the board. She'll lay eggs in tree holes, tires, containers. I found her in ashtrays before. So her dispersal strategies is quite similar to the last mosquitoes we've been talking about. Egg desiccant retolerant. This is one of the things that from an evolutionary standpoint is pretty impressive with these mosquitoes, that they can move in and around and have these delayed response times for hatching, and then also can resist desiccation and propagate itself over time to pop up one day and be like, hey, we're here. I know it took me a couple years to get here, but now I'm here. Ports of entry are a big deal for Aedes japonicus. I was just visiting Savannah, Georgia, where they have a port there, and they were showing me the site right outside the port on the other side of a dredge site that the Army Corps of Engineers manages, that they found Aedes japonicus in the middle of nowhere. Pretty interesting. So collection methods, tough. She doesn't like coming to CDC traps. The males will. Larval surveillance is key. She will go to octanol-based type trapping mechanisms. Gravid and oviposition traps are ideal with collecting this mosquito. She will come sometimes to Biogen's, the BG Sentinel traps. Aspirations are amazing. Wide bore, Nashi aspirators, the Procopac aspirators to get into tight crooks and crevices and things like that work amazing for collecting Aedes japonicus. Host biting preference, wide ranging. She likes those crepuscular type times. She does like the fringe of places. She likes cooler places, shade. She will come out and feed on you. She is opportunistic, as with this photo. She will feed on Sciuria type animals, marsupials, us, large mammals, and birds. But check out that pathogen transmission platform, wide ranging. Now, 60% of these are dissemination events in laboratories, and some of these are natural occurrences. In her native range, Japanese encephalitis virus. She is known to be infected with Lacrosse virus in North America, West Nile virus in North America, Cache Valley virus in North America. Rare occurrences. Let's dig into the morphology. I'm super excited about this mosquito. I've been following her for quite some time. She's a medium to large mosquito, and when you see her, you can't unsee her. She's very opaque across her vibrancy. Even an aged mosquito does pop. She has very, very wide bands on her tarsi. When you look at this mosquito initially, you do see this brown, goldish kind of hue to her, with an ivory kind of pop on the lateral view of her thoracic body. The one thing I want you to take note of are the wide bands on number 2. These things are very wide. When you look at, let's say, an Aedes aegypti or like an Aedes taeniorhynchus, these bands are very wide and very distinct. So let's take a look at one of those legs on number 3. We're going to be looking at this femoral character, and we're going to dive into this a little bit more. But this is something that's an indicator within the complex of Aedes japonicus mosquitoes. There are differences within the orientation of shape and shading of this band on the femur. So let's take a look at these golden stripes that are distinctly lyre shaped. So far we've had a couple of mosquitoes that we've been looking at that have lyre-like shaped markings. Well, within the Japanus complex, there are lyre-like shaped markings. They are golden. They're very distinctive. The one thing they do have is a medial thick line going right down the middle of the mosquito, and and 2 submedian stripes that come down parallel right above the curvature of the lyre-like shaping as it goes down the lateral side of the scutum towards the end of the scutellum. I need one moment. Thank you. So this is very distinctive within this complex, and we're actually going to look at not only this mosquito, but we're also going to take a look at a mosquito called Aedes togoi and a mosquito called Aedes coriacus. I'm currently working on building a slide deck for Aedes togoi. I've got some specimens back here. We haven't processed them yet. But one thing I want to talk about is within this complex, there are some similarities within the larvae. When we look at the larvae, uh, I want you to really take note of number 6. And one of the reasons why I'm talking about the larvae is because this is very different from every single larvae that you're going to find in a container in North America, Mexico, or Canada. She has a linear orientation of fan-like setae that go across the top of her head. And they're very fan-like. You are going to see this on Aedes togoi and Aedes coriacus. You're also going to see something as in number 7, where the saddle is conspicuously spiculated on the fringe of that saddle as it's coming up towards setae S1. You're going to see these spines that are very pronounced and coming off the fringe of that saddle. All right, Aedes coriacus coming to a container near you. This mosquito is similar to its pathway into North America as Aedes japonicus. Her native range is China and the Korean Peninsula. She has had a very interesting journey across Asia and into Europe. She does habitat a wide range of different types of water columns. When you look at her expansion into Europe with these green instances of presence throughout, um, I want you to look at the North American map of the United States. And you see those red dots all in and around the northeastern side. That's like New Jersey, Rhode Island, and these port type cities there. This is the same introduction site as Aedes japonicus back in 1995. This is a predictive map that they're predicting that if this mosquito does come into the United States, this is most likely the area that it would land. Her larval habitats, catch basins, temporary surface pools, artificial containers. You're going to find her throughout. She has an urbanized affinity for associating with us. You can find her in cemeteries. Her dispersal strategies. I think we're starting to see a trend, right? We're seeing a trend with these container inhabiting mosquitoes. We're causing our needs and resources from around the world are attributing, we're basically being taxi service to these invasive mosquitoes. Now, collection methods, BG traps, just like the last. There is some mentions of mosquito magnets, but those things aren't even available anymore. Gravitrap she will come to. Her range of invasiveness across Europe started in 2008, which is kind of interesting, where she popped up in Belgium and kind of moved around Europe and then kicked out to the west side and jumped into Kazakhstan. It's kind of interesting through Ukraine. Her host biting preference, us, large mammals, sometimes birds. There's a hypothesis of lizards. There was some possibility of blood meals showing lizard DNA. Now, she does have affinity for Japanese encephalitis virus as a pathogen that she can transmit. Also, dog heartworm. So this mosquito, I'm still trying to get specimens. These are from the Smithsonian. They were gracious enough to give us specimens to take a look at. So this is where we're going to get into like some similarities, right, and differences. So we're looking at Aedes japonicus. Now we're looking at Aedes coriacus. This mosquito, when we're looking at number 1, this is a specimen that's been in curation in the museum for quite some time. So there is a drab look to her. In her natural environment, if she had been around for a week or two, we're collecting her, she would have that vibrant black look with the ivory scaling pattern. She would really pop out. But what I want you to pay attention to is not the vibrancy or the dullness of the specimen that's pinned here. But what I want you to look at is the orientation of scaling patterns on that lateral view of the thorax. There's some similarities with Aedes japonicus. And then I've done some interesting zooming here. So look at the zoom um, in number one, where we're looking at that subspiracular. No, we're looking at the tarsi, and we're looking at these knee joints here. You can also look at the bubbles on the big picture of number one, and you can see that that pop out on the leg, and you'll see that scaling pattern on the femur. This is something to pay attention to. So when we're looking at number four, and as we're looking at number one, you see the circle down at the bottom. What's interesting about this is that all legs have basal tarsal bands. The femur, the tarsi 2 with the anterior and dorsal surfaces are white apical. The tarsi 3 is white banded basally. And then you're going to have a kind of a dark tip on that fifth tarsi. So number three, I've got some red lines to help you out. This is an older specimen. Those lyre-like markings are a little bit difficult to see on this photograph here that was given to us. But what I want you to take note of is the lines of the lyre-like markings that are going down, and then that break at the bottom of the scutum, which is reflective of Aedes japonicus as well. Now we're going to get a little bit sticky. So now we have three mosquitoes. I know I don't have a slide on Aedes togoi, but they all are similar looking. Aedes togoi, there is a population of them in the Northwest, Oregon, and Washington states. Recently had the opportunity to collect some of those back in February, coming to a slide near you. So these mosquitoes all look similar. They do all look different. And the similarities that we want to look at are those thoracic views where the patterns are somewhat similar. We're looking at those banded legs that have wide bands all the way down, the dark proboscis, the dark wings, the golden-like stripes that are actually on the scutum. So let's dial in on these character traits. So now we've got them side by side. When we're looking at the scutum with the golden stripes, you have these distinctive lyre-shaped stripes with two submedian and a median stripe. In the orientation on some of these, some are a little bit wider. In the middle, those lateral medial bands are pinstriped in some instances, but when we look at Aedes coriacus, that medial band that's coming down is quite wide. The submedial bands are quite wide. But you see on the Aedes coriacus, coming around the scutal fossa down towards the back end of the scutellum, you can picture that lyre-like marking. It's not so pinstriped as japonicus or togoi. There are some differences but similarities all the way across. This is one of the reasons why I put these mosquitoes side by side. Now let's dig into what Tanaka 1979 really wanted us to dive into with this complex. So there's similarities and differences within other structures. We want to look at the middle femur. There's an interesting text out there back in 1979 that got revisited by Cameron, and we'll go over that. But the origin at the middle of the femur, take note of these shapes. So apologies for coriacus not having an arrow. For some reason, it's disappeared. But I want you to pay attention to the point of the arrow, the differences of the ending of the shaping of the orientation of scales on the inside of that femur. When you're looking at japonicus, it's got almost like a pointed finger. So does Aedes togoi. But when you look at Aedes coriacus, it's kind of blunted and has a notch at the end, kind of like this. So when we're thinking about that insertion site, where you have the femur coming up into the trochanter onto the body of the thorax, The scales are not touching the insertion on Japonicus. They are touching the insertion on Togoi, and they are touching the insertion of the thorax on Coriacus. So there's differences throughout. The orientation of these femoral characters have been tested through phylogenetics of Aedes japonicus across the board, have been looked at from every single species within that complex that have similarities, and the difference of the orientations of the scaling patterns on the middle femur are very distinctive by species. If you have mosquitoes and you want to make sure which it belongs to, you can look at these femoral characters. This is basically a go-to to decide which species within the Japonicus complex that they belong to. These femoral characters are amazing at deciding who's who and what's what with this complex. I hope that's helpful. All right, now we're pulling out of container inhabiting mosquitoes. Aedes pertinax. This is one of those mosquitoes where, like, how'd it get here? Why is it here? There's some ambiguity of Aedes pertinax through its taxonomic history of identification, but that's okay. Its actual distribution is widely unknown. Good for everybody. Right. Native to South America and the Caribbean. Invasive to Florida currently. The expanding range of this mosquito, have not updated this map. Working on that currently. But the West Coast, she's also found in Indian River. That was the initial report. I have heard of reports of central to West Coast central counties finding Aedes pertinax in their collections. Her larval habitat. More research is needed in this area. Coastal flooded woodlands, sometimes maybe some brackish water possibly, temporary permanent grasslands, woodlots prone to flooding, was found in freshwater sites, non-tidal, rain-filled depressions. Dispersal strategies, more research is needed in this area. No flight distance is recorded for this species. Hypotheses for her dispersal in this area, major storm winds driving dispersal from her habitats in the Caribbean. Collection methods can be abundant in CDC traps and BG sentinel traps. Larval collections, fairly apparent through dippers. Host biting preference, guess what? This is why I have the question mark with the little guy in the bottom right-hand corner. We need more information. Pathogen transmission, need more information. But the interesting about this thing about this mosquito is her morphology. Here in North America, outside of Aedes pertinax native zone, where she's now spreading throughout the entire state of Florida, she looks exactly like, in some instances, Aedes atlanticus, and then her sister species, Aedes tormentor. The folks that have ID'd in North America know about Atlanticus tormentor and the debate between this mosquito and separating it from one another. Now we're adding another mosquito into the mix that they look similar to. So when we're looking at the lateral view, number one, this mosquito has interesting markings on its abdomen. No complete banding, these lateral spots going all the way down like triangles. The intricate patterns on the mesocat episternum, the mesoperion, and then we can look at number 3, 4 at this interesting stripe that's going down. You see how it's kind of pinstriped at the top but gets wider at the bottom. I need you to move to number 6 and take a look. Absent, narrow, variable, disconnected. These are all mosquitoes that pupated in captivity. These are not mosquitoes that were rubbed from a fan. So there's high variability in the type of medial lines that are actually going down, and these mosquitoes were tested and verified to be Aedes pertinax. We're not going to go into 7A, 7B, or 7C into the larval characters. But what I want you to look at is the longitudinal stripe on number 5 versus the longitudinal stripes in number 6. Kind of get that in your mental Rolodex there. that visual platform you use when you're doing ID work in your scope, the continued occurrence of seeing scutal characters that are similar, and how are you going to decide who's who and what's what? So let's challenge ourselves. This can be tough. We're looking at Aedes pertinax. She has a medial line. Aedes atlanticus, and this is a fairly decent emerged specimen. And then we're going to be looking at Aedes tormentor that's rubbed by a fan. But you can see the remnants of scales on that Aedes tormentor. When we're looking at Aedes atlanticus, you're seeing similar type markings. But when you're seeing that Aedes pertinax, what are the differences here? What happens if the Aedes pertinax scutal characters are rubbed off? What happens if all three mosquitoes you've got in the same net, everything's rubbed off all the way through? You can take some indicators and do some troubleshooting work with these three mosquitoes. Currently, we are working on occipital type scaling patterns versus Aedes pertinax, Aedes atlanticus, and Aedes tormentor. We're working on scalings up to and connecting or disconnecting from the compound eye. This work is currently ongoing. And so coming to a slide near you, the challenge currently is a real one. This is an ongoing project of identifiers across the state of Florida looking at some of these character states, making notations about these differences after you might have rubbed off specimens. So this is real-time work. Pretty impressive. Aedes scapularis. We have this mosquito in this slide. I'm going to check my time real quick. We have this mosquito in our slide deck as a re-emerged mosquito coming into the peninsular area of Florida. Her story started in North America 80 years ago, has been non-present, all of a sudden popping up as of 2025 through some observations in the peninsular area of Florida. Her native range is basically South America, the Caribbean, and Central America. Her expanding range currently is in South Florida from the Florida Keys and Monroe County all the way to the west up to Sarasota County, all the way over to the east to St. Lucie County. There's a current project going on coming out of FMEL about the expanding range of Aedes scapularis, even in some of the central counties. I'm not going to say who they are. They're waiting on the publication to come out. But this mosquito is expanding and moving north. Her larval habitat is not really containers. Sometimes temporary semi-permanent pools, freshwater such as swamp margins, sometimes crab holes in the literature, overflowing waterways and rain pools. She's an opportunistic floodwater mosquito that will overposit in the muck and things like that. She will lay eggs prone to flooding. Her dispersal has been hypothesized to occur by aircraft, movement of plants and soil where eggs and larvae may have been present. Her collection methods are you can catch her between CDC baited CO2 traps, light traps, and Biogent Sentinels, BG Sentinel traps. With this pie graph, you can totally see that she's an opportunistic feeder. What does that mean? There's a common trend here. If you're an opportunistic feeder, your pathogen platform for transmission is fairly wide. Some things here that are interesting are Pouchet virus. That's been in the news lately, and it's been the buzz over the last year and a half or so. But one thing that's kind of troubling here is yellow fever virus, right? That's not good for us. So let's take a look at this mosquito, and we're going to take a deep dive on this mosquito to another mosquito that is similar to. But let's take a look at the lateral view. Number one, what's the similarity that we are viewing here that we noticed with Aedes pertinax? Incomplete, no bands on the abdomen, and then we're looking at those triangle patterns down the lateral side of that abdomen really pop out. So when we're thinking about some of these floodwater Aedes, like your your Aedes duprei, Aedes tormentor. You're going to see these types patterns on the abdomen. So is that really a character state that we can use maybe from other sets of Aedes? Yes. Other floodwater mosquitoes, sure. What we really want to take a look at is number 2 on the abdomen. This is very pronounced on from the target 4 to 6. You're going to have, or to 7, you're going to have these light colored scales immediately that taper from the last target all the way in. It tapers to this nice little line. I'm actually working on getting a photo of this character. I'm just waiting on the email. So apologies on the line drawing. But let's take a look at number 3. This scutal character. This is way different from what we were seeing with Aedes pertinax and Aedes tormentor and Aedes atlanticus, where this medial patch is fairly wide and almost encompasses the entire scutum. This is a very pronounced patch of ivory to yellowish scales that you'll view. And then we can move along to number 4, where the occiput is clothed medial with white or colored scales, all the way encompassing all the way up to the compound eye. So when you're looking at it from the compound eye all the way down, you can kind of see what I'm talking about. Go to number 1, view the compound eye, go through the occiput, the scales there, and then follow that all the way down to scutum. It's very pronounced. What I really want you to take a look at is number 6. So number 6 is really going to show you something that's interesting with this mosquito versus Aedes infirmatus, where we're looking at basal and apical bands on the hind tibia. So this is where things get kind of interesting between these two mosquitoes, where Aedes scapularis and Aedes infirmatus have serious similarities across the board. Look at the lateral views of both of these mosquitoes. When you initially take a look at both of them, you're like, whoa, holy crap, they're the same. But let's take a closer look at this hind tibia. On Aedes scapularis, it has basal and apical dark bands. Almost, it looks like the middle is like banded pale. Let's take a look at that hind tibia on Aedes infirmatus. Right. It's dark scaled in its entirety. There are no bands. There is no pale band. There are no splits in scale colors. This is indicative of Aedes scapularis, where there is a band. I was just currently, I almost slipped, at a county in South Florida where they invited me in to verify their collections, which is now going into a paper. That tibia character is great for separating these mosquitoes. But let's take a look at some differences between Aedes scapularis, Aedes infirmatus, and Aedes convalescens. Now, we haven't really talked about Aedes convalescens, but when we're looking at these mosquitoes and we're looking at similarities and differences of scutal characters, the species with the medial longitudinal stripe extending from the anterior margin is about 2/3 the length of the scutum. So this is the similarity. What we're looking at here is interesting, is that Aedes scapularis has a wider band. The only other mosquito that you could contribute this wide band to would be Aedes duprei, but Aedes duprei is a teeny, teeny, tiny mosquito. It's very small compared to these medium-sized mosquitoes. But these characters can be rubbed off. So let's look at that secondary character state of, I'm going to go back and show it to you again so you can't unsee it. The band on the hind tibia at Aedes scapularis is very pronounced. With Aedes infirmatus, it's an easy decision. Once you see it, you can't unsee it. I hope you enjoyed that run. All right, the next thing we're looking at is Anopheles stevensii. Why do you ask that we're looking at Anopheles stevensii? So if you remember at the beginning when I was introducing beacons, we shine the light, right? We shine the light of something that may occur. So we've already looked at a couple mosquitoes, Aedes vittatus, we've looked at Aedes coriacus. We're also looking at Aedes nondescriptus that's on the west coast. It might come down I-10 to the southeast. So we take this proactive view on mosquitoes. And what's interesting about this mosquito is her adaptability to containers. Her native range is India, Arabia, a little bit of Southeast Asia. Um, she is a malaria vector that's in a container. She's adapted to um, urban environments. This has flipped the script of the malaria story in Africa that used to be this rural, sub-rural type transmission cycle that's going on where your city centers had introductions, but it wasn't being spread. Well, guess what? Now it's being spread. This mosquito came upon our radar based on her very fast track across the continent of Africa from 2012 to 2022, went from one side to the other. And guess what? It's in Ghana. And currently, that country has ports. And what is one of the main things we've been talking about with dispersal strategies and container habiting mosquitoes? There's a common trend. If it can get into a container, it can get here. So, these anthropogenic commerce shipping distribution lines are really exasperating the possibility of this mosquito coming. So we wanted to shine a light on Anopheles stevensii as a container inhabiting Anopheles malaria vector. Larval habitat, wells, cisterns, ornamental ponds, fountains, pools, stream margins. She will slide from containers into things like catch basins, seepage canals. You can collect her through larval collections, pit shelters. If you notice the tent, there's an actual cow inside that, host baited traps, landing rates. Does anybody want to do landing rates with a malaria vector? I don't, right? So, I mean, it can be fairly ambiguous. Host biting preference, large mammals, humans. It's endophilic urbanly, but in rural areas, it's exophilic. So it has an adaptability range for host preference, inside, outside, based on where it's at. Pathogen transmission, plasmodium. Oh, crap. Chikungunya too. All right, let's take a look at the orientation of this mosquito morphology. Hopefully I've raised some eyebrows with that beam of light. Anopheles stevensii. So in my class, when we're teaching this in face, I put this mosquito together with Anopheles gambi and Anopheles crucians. I don't have those slides currently, so we're just going to go over the morphology, the adult female morphology of Anopheles stevensii. I haven't really created those slides currently. So number one, the female adult. So we haven't seen an Anopheles today, right? I mean, we've been mainly looking at Aedes. So let's look at this mosquito. She's fairly ornate versus some mosquitoes you will see here in North America. Her scutum is obvious pale scales, and her setae scutofossil with pale scales. She's got this on number 3. The palps have pale bands, but they also have this smattering of scales going throughout. 3A is the head with an erect, entirely pale scales. And when I'm talking about erect scales, I'm not talking about setae, right? I'm talking about these forked pale scales that come up. They're kind of forked like that. And they kind of, let's see. Oh, yeah, they kind of go down in a position like this. They're very erect. The wing, pale spots present on nearly all veins. But what I want to really take a look at is the anal vein, where vein 1A with 3 dark spots all the way across. Check out the orientation of the pale scales versus the dark scales. Now let's take a look at number 5, the femur and tibia with small pale scales throughout, the tarsi with narrow pale bands at the joint. A very ornate mosquito. All right, we're hopping into Culex coordinator. Its native range, South America, Mexico. Its non-native range from Texas to the East Coast has taken like 80 years, like 100 years for it to move across. Some recent reports has shown its adaptability through different environments in the Southeast. Dr. Bill Sams did an amazing work on the southern distribution currently of Culex coordinator. She is considered a container having mosquito, but she will lay her eggs in roadside ditches, animal water troughs, forest ponds, rock pools. She's highly adaptable to artificial containers, and that's how she's moving from place to place. You can find her in tires. Car tire transportation is thought to be the most important dispersal mechanism. So this is a Culex that's being dispersed in the ways that we're seeing the Stegomyia and Aedes japonicus and things of that nature. She will come to CDC traps, BG traps, gravid traps, larval surveys, aspirations, predominantly nocturnal species. She's opportunistically will feed on birds. She'll feed primarily on large animals like horses, white-tailed deer. She'll also feed on us. Pathogen transmission, St. Louis encephalitis virus, West Nile virus, and some instances Zika. This mosquito is fairly interesting. It does look like Culex tarsalis. In my class, I put them side by side so the students can make their differentiations. She is somewhat drab. Culex are kind of drab. This is a more ornate type Culex. She has you're looking at number 2. You have this like half band on the proboscis. Number 4 is that the hind tarsomeres on the legs are ringed with distinct pale bands and apical bands all the way across. The one thing larval-wise, this mosquito at the tip of her siphon, and this is her namesake, has these spines coming off the tip of the siphon, and that's what dictates the word coronator. It makes it look like she has a crown. They're conspicuously spiculated and spiny at the top of the siphon. We're moving on to Culex declarator. Here's another mosquito that its native range is South America and excuse me, Central America. She comes to us through Texas into Florida and into the Southeast. Her current range in Florida is quite a few different counties, ranging from the Panhandle all the way to the southern end. Her collection methods are CDC traps, resting boxes. Her larval habitats, she can bounce between rural and suburban habitats from tropical to subtropical. She likes this moist, broad leaf forest, tree holes. She can be close to temperate ponds, swamps, and small dams. She will also get into containers, artificial containers. She has non-human mammals for biting preference, birds, less frequently humans, excuse me. Pathogen transmission, we're looking at St. Louis encephalitis virus, West Nile virus, and dog heartworm. More research needs to be done on her dispersal. So this is another drab Culex, but there are some interesting things we need to take into consideration here. Number 1 are these dark brown narrow scales with lateral brown spots. So you're going to see some of this intungamental spotting on some of the Culex we're going to be looking at. That create a stripe pattern and lateral small patches of white scales. So really look at number 1 and kind of take a look at that. Number 3, we're going to see these white lateral patches going down the lateral side of the abdomen. Take note that number 2, the proboscis has a long pale area on the lower surface. It kind of remnants of Culex coronator. The one thing with this mosquito is number 5 is that the femur and the tibia have pale tarsal segments. They can be dark scaled or with very narrow white bands. And when I say narrow, they are very narrow. You really have to dial in on the segments of the tarsi to be able to see these bands, but they are there. We are moving to Culex interrogator. Culex interrogator, guess what? This mosquito's native range is Central America. It's coming to us from Texas. Texas is definitely an entryway for non-native mosquitoes into the United States. Its current expansion, currently as of 2000, I think 23, is over about a third of the counties in the state of Florida, wide ranging from the western panhandle to the northeastern side up near Jacksonville, all the way through a smattering of counties throughout the central and southern areas of Florida. Her expansion has been attributed to containers, likely facilitated through mosquito-infested containers along major highway routes. So permanent, semi-permanent habitats, including natural and artificial containers. So here's another container inhabiting Culex, but also opportunistic ovipositor, where she won't only just layer eggs in containers. Significantly lower abundances as compared to like a Culex nigerpalpus or Culex coordinator collection-wise. Adult collections are rare. You do see more larvae. Overtraps are great. Gravitraps are great. Infusion traps are amazing at collecting this mosquito to collect egg grafts. Host biting preferences, and this is a pattern that we are seeing with some of the mosquitoes that are being introduced. More surveillance and research is needed in this area for a host biting preference. So if anybody's got any blood-fed Culex interrogator, please contact us. So it has been collected in some dog-baited traps in Mexico. It is associated with dog heartworm, also associated with West Nile virus, indicates bird blood meals. But pathogen transmission, this stuff is fairly ambiguous. Her role in these things are quite unknown, and there just needs to be more research in this area, although with dog heartworm, it's pretty much indicated. So this is a small mosquito. It's a small Culex. Not as small as your subgenus Melanoconian, like your Culex erraticus or your Culex iolambus, but definitely the coloration of these mosquitoes are more brown versus the grayish of the subgenus. The hind tarsomeres are entirely dark, or with narrow pale bands, basally on the tarsi. So take a look at number 4. Where have we seen this before? Culex declarator, those dark integumental spots surrounded by patches of white scales on the thoracic body. When we take a look at number 3, the wing length is 3 millimeters or less. We will talk about some things on number 6. It does have those dark integumental spots, and then this will really pop out on this mosquito. But what makes it tough is that this mosquito's abdominal bandings are similar to Culex quinquefasciatus and Culex restruens. In cell number 6, cell R2, that radial vein coming across, and where it branches off is about 1.2 millimeters or less length of the vein of R2 plus 3. It can get complex. So let's take a look at 3 Culex mosquitoes side by side. In my in-face classes, I have all 3 of these mosquitoes side by side together. Take one glance and let me know if you're looking at differences or similarities. When we're thinking about these abdominal turgets and the bandings on them, this is what I want you to focus upon. Oh, wait, there we go. We're blowed up. So let's take a look at Culex interrogator. We have a lateral view of the interrogator. We have a dorsal view of the interrogator. What I want you to pay attention to is turgets 5, 6, 7, and 8. When we're looking at Culex interrogator, we're seeing almost like a moon, almost a moon shape. There's almost a curvature. We're looking at quinks, there's a curvature. We're looking at restruens, it's pretty much a linear flat line. You start having a curvature on 4 with restruens. Then you start seeing like these little scooped out areas that aren't so concave. They're more concave than convex versus Culex quinquefasciatus and restruens, When you start looking at Culex interrogator, they start to get a little bit more concave on Culex interrogator. So we see these similarities between 4 and 6, between all three, and then you start seeing distinct differences at 3 and 4, and then 7 and 8. It's fairly interesting to take a look at these. But wait, it gets worse. Let's take a look at the wings, right? First initial viewpoint on these wings, they all look similar. They are dark scaled. The fringe are dark erect scales. These veins coming through from the anal vein to the cubital vein to the radial vein, all the way up through, they're all dark scaled. The differences between these is the size and the length of these. I hope you can see my mouse as we're going through this wing vein here, and then the branch here, this cell length versus this length versus the length of the entirety of the wing from the subcostal area all the way down to what we call the fringe tip. The lengths of these is what determines the species differences between Culex interrogator and Cuenca fasciatus or pipiens and Culex restruens. It can get fairly boggling down when you're diving in. I suggest that if you're having troubles with abdomens and you're having trouble with these integumental spots that you're going to see on Cuenca fasciatus and interrogator, restruens pulls you right out. Those spots aren't even there. Pull that mosquito out. It's gone. So guess what? Wow. Now all we have is Culex interrogator, Culex Cuenca fasciatus to look at, right? So you pulled restruens out. Now we're going to start looking at these abdominal targets. The interrogator is going to have more banding on those last targets. There's going to be more pronounced pale scales. So before you start doing wing measurements, take a look. So let's dive in at these differences. You're going to see integumental spots on interrogator. You're going to see pronounced on Cuenca fasciatus in most populations. The mesocat, episternum, mesoperion were without dark integumental spots, but you are going to have scale patches with 6 or more on all three. All right, Culex lactator. This is another Culex mosquito that's been introduced into the United States. Hey, we're starting to see some similar patterns here. Its native range is South America, Central America. Its current expansion is into the southern peninsular area of Florida, encroached into Broward County, Miami-Dade, Collier County, and then a record on the border of Lee County and Hendry County in South Florida. Her larval habitats, catch basins, stormwater issues, groundwater, rainy seasonal April through July, flood water. Can be considered a residential harbor in some of these areas that collect water. It might be impossible to determine the point of introduction for this mosquito. She's not a container inhabiting mosquito. So that introduction and geographic chronology of dispersal within Florida is almost certain that Culex lactator is spread within Florida beyond the introduction point. What is that dispersal? It's tough to say. So there are some hypotheses. Adult collections are rare, but have been observed using gravity traps, CDC baited with CO2, and through mostly larval collections. Guess what? Host biting preference. We need more surveillance and research in this area. Pathogen transmission, more surveillance and research is needed in this area. Let's take a look. This is a really interesting looking mosquito, Culex lactator. So I've got some things, red arrows going to tarsi. I've got red arrows going to the thoracic area. We're going to blow those things up. Take a look and get oriented on the lateral view of this mosquito. A lot of these Culex are drab looking mosquitoes. They all kind of look similar when you're looking at them through the scope. But number one, there are faint basal and apical pale bands on the hind tarsomeres. The complete or incomplete band on the proboscis is present. So it can be confused with the incomplete or complete bands on between Culex incornator and Culex tarsalis. But on number 4, there are apparent patches of pale scales on the upper and lower, the mesocat, episternum. When we're looking a little bit deeper at this, we're looking at patch of pale scales on the mesoperion, but let's look at 5A, right? The arrows should be also going to this version here. 5A is supposed to have an arrow here. So we're looking at these light, dark, intergeminate spots. It gives that banding that we saw on Culex interrogator and Culex declarator. We're starting to see a pattern with some of these Culex mosquitoes. The one thing that really stands out with this mosquito is the length of the antennae are longer than the proboscis. Take a look at that proboscis from 2, view on number 6, and it almost has a bulbous kind of chunk at the end of that. And we've kind of hypothesized that it has this kind of appendage on its proboscis for feeding on birds and getting through the feathers and getting down inside there. All right, Culex nigerpalpus. Who thinks that this mosquito is a non-native mosquito? Is it invasive? It definitely has an impact from an arborvirus perspective. Its native range is the southeast of the United States. You can't find this mosquito in other places, but it is expanding. It's expanding out of Florida. It's expanding into other states like Alabama, South Carolina, and now all the way up into Virginia, up into Suffolk, they're finding this mosquito. So this is another instance of where mosquitoes are actually coming out of Florida, and we're going to have another example of that with Mansonia, where this mosquito comes from permanent transient pools, freshwater spot, swamps. Its flight range is interesting, flat 5 kilometers. How is it moving? It's not really considered a migratory species, but it is expanding. Anthropogenic introductions cannot be ignored, increasing temperatures as things progressively get warmer. Collection methods, a wide range of collection efforts to collect this mosquito. Her host biting preference, mainly birds, reptiles, mammals, hypothesizes a major zoonotic transmission contributor for St. Louis encephalitis virus and West Nile virus. Culex nigerpalpus is a nondescript Culex mosquito. She's kind of drab looking. Some people say she's kind of boring. I think she's interesting looking because of the absence of things. So she is a medium-sized dark brown mosquito, dark scaled. The wings are dark scaled. The proboscis is dark scaled. She has these interesting acrostitial setae that go down the medial line. They're very pronounced. They kind of poke out in different random points down the medial line. Let's look at number 2 on the thoracic area. No scales present on this current specimen here. If they are present, it's less than 5 pale scales on the mesoperion. As we've seen with some of the other Culex mosquitoes, the triangle-shaped pale scale patches, there are no bands across the target, as we're seeing with some restruans and Quincke fasciatus. This is a very nondescript mosquito. Everything is pretty much brown scaled, dark scaled. There's not a lot of orientation. All right, now we're going to be moving into the subgenus of Culex, Culex panicosa. There's only a couple of instances. This mosquito is fairly rare in Florida currently. Its native range, South America, Central America, some spots in the Caribbean. She's found in a couple of different places currently in South Florida. You can find her in canals, ponds, wetlands. Water lettuce is associated with this mosquito. She has adapted to human environments, so these man-made water remediation areas, she will inhabit those. Could have been mechanical dispersal strategies through source reduction, remediation of waterways, wide range of different collection methods from aspirations, wide bore aspirations, all the way down to CO2 baited CDC traps. Wide range of hosts include humans, including reservoir hosts such as lizards. Her pathogen transmission, she's a suspected vector of Venezuelan equine encephalitis virus. Here's a pretty cool photo of this mosquito taken by Dr. Nathan Burkett Cadena out of the Florida Medical Entomology Laboratory out of the University of Florida. We're very appreciative to get this photo. And then here's some amazing photos. These photos are taken from the subgenus Melanoconian identification key that's in the Journal of American Mosquito Control for these subgenus within Culex in the Southeastern United States. If you don't have it, I suggest that you pick it up. So we're looking at Culex panicosa on the right. On the right is A. Culex nigropalpus is B. So we can see some of the coloration differences that are going on. The presence of acrostitial setae. Excuse me. When we're looking at Nathan's couplet photos from the Southeastern Mosquito Control, the Southeastern Mosquito Identification Guide, those acrostitial setae are present. The occiput, the erect pale scales, the erect dark scales, versus the flat pale scales. But we're also looking at basal pale bands across the board. When we're looking at Culex panicosa, she is a very small species, mostly dark scaled. Those acrostitial setae are absent, dark brown integumental. So when we're looking at number 4, we're looking at these plural integument scale, pale yellowish contrasting on the scutum. And if you're looking at the contrast of the dark coloration of those spots versus the coloration of the thoracic body, it's very pronounced. So remember, with these mosquitoes, the acrostitial setae are not present. So number 5, these are distinct spots on different structures along the thoracic area. The postpronotum has a spot, the postspiracular area has a spot, and the prolorar knob has a spot. This next mosquito is a tree hole container mosquito that's found in Texas. It has expanded, or at least it's been noted in a new county in October 2022 outside of a previous collection from 1962, Haemogagus equinus. Its larval habitat are tree holes. It is a medium to large species. Its dispersal strategy is unknown. Collection methods, I'm guessing turkey baster. This species can be collected from filled tree holes, especially after increased rainfall. Her host biting preferences is birds, mammals. She's hypothesized as a zoonotic transmission contributor. Those transmission cycles are wide-ranging, from dengue, yellow fever, possibly some other pathogens, but not really has been evaluated through her vectoral capacity. Haemogagus equinus, what do you think of this mosquito? Pretty ornate. When we're thinking about this mosquito, look at the lateral view here. There is a lot going on with this mosquito. It has a very tapered abdomen, very long legs, kind of reminiscent of between a Toxorynchides and a large Psorophora, like a Cyansins or something like that. The pre and post spiracular setae are absent. Should key out somewhere pathway on as Aedes. Abdominal and scutal scales are metallic blue, violet, and silver, and almost makes it look like there's a stripe going all the way down through. The occiput has broad, flat scales of metallic blue, violet, and silver. It's very popping. So when we're looking at number 3 and 4, and we're looking at the scutum scales, look at that ornate band. It really pops. It's a really beautiful mosquito. The silver on number 5, the silver scales extend from the scutum to the coxa without interruption. It continues all the way up through that pattern, forming a single vertical band. And then there's some larval characters are described in Darcy and Ward. This is probably one of my favorite mosquitoes currently. She's just really ornate. I hope you guys are enjoying this slide. All right, so I said we were going to return to mosquitoes coming out of Florida, invading everywhere else. So Mansonia titulans. We all kind of know Mansonia. They are associated with aquatic plants. They have modified siphons that attach to the roots of aquatic vegetation to get their resources. Their native range is very wide across the globe. Its invasive range is very wide as well. You can see this pattern of expansion with this species. So we can just expect that Mansonia is going to expand. Recently detected in South Carolina, the last 20 years, migration over 15 counties in Central and North Florida has been apparent. Permanent bodies of water associated with water hyacinth, water lettuce in some instances, dispersal strategies, max flight distance, known to fly several miles in search of hosts. Larvae may be transported through ornamental aquatic plants. CDC light traps for collections, BG traps, emergence traps placed over larval habitats, larval collections best done by removing and washing the host plants. In some instances, you can have modified dippers that have screens, and you use a scraping method. Host biting preferences, they are generalists. They feed on a combination of mammals, birds, and humans. A wide range of pathogen transmission, as we can expect from her opportunistic host preference, right? So this is also a pattern that we are seeing. All right, Mansonia titulans. She is a medium-large species, speckled scales throughout. So we started out with a speckled mosquito. Who all remembers that? Right? The ornate patterns on Adiomyasquami penis. And so what I was talking about, the ornate patterns on Mansonia and Colquittida are reflective here. So when you look at number 3, you can see the ornate pattern of dark and white or pale broad scales on the wing. Back to the lateral view of Mansonia titulans, it's a medium to large species, speckled scales throughout. So you'll see speckled scales, bands on the abdomen, over the wing, on palps, on the abdomen, speckled scales on femurs, and the abdomen is blunted, not tapered, not rounded. It also has a banded proboscis, banded tarsi, and banded tergus on the abdomen. So number 4, those tarsi pale scaled bands are below the joint. Now, this is where things get interesting. Remember how we were looking at those container having mosquitoes, and we were looking at palps, and we were looking at flagellomeres, and we were looking at pedicels and clypeus? Well, guess what? Here we go again. Palps almost half the length of the proboscis with this mosquito. So we're actually looking for the length of something compared to the length of something else to make a determination. That's really interesting. We're also going to be taking a look at flagellomere 1, so dial in on that pedicel on number 6. The flagellomere arises from that structure on the interior of the first segment of the antennae, the flagellomere 1. There are up to 5 dark scales present. They're not on its sister species. We're going to take a look at that. All right. We're going to take a backtrack to number 2. On that blunted abdomen, on the fringe of that abdomen, it's really difficult to see it on this slide, most likely. There are spiniforms. Spiniforms are a rigid setae structure that is pointed, that gives rise off the fringe of the end of the last tergit. It almost looks like a comb, but it's very rigid. And they're along the entire fringe. A lot of times these spiniforms can be broken off. Take note of number 8, the modified siphon, and then you can see the attachment of the larvae onto the root of a plant structure. So we're going to do a little comparison. Let's take a look at Mansonia titulans and diari, its sister species. The last abdominal segment with a row of black spines. In some instances, those things can be removed. On Mansonia diari, those are not available whatsoever. So that's 2 things you can totally remove each one from the other. Dark colored scales medial on the first antennal segment. Now I've blown this up so you can see it, and this diagram is from the Mid-Atlantic Guide by Bruce Harrison et al. 2016. This is a character that he found where you can zoom in on that first antennal segment, and you'll see these 4 to 5 dark scales. When you look to Mansonia diari, that first antennal segment uniform in coloration, so there isn't any dark scales. It's going to have brown all the way throughout. All right, we're kind of funneling down. What time is it? Are we over?
No, not yet.
Okay, cool. Thank you. This is an interesting storyline here, and so it is at the end. Raise your hand if you've ever ID'd an Aedes vexans, right? It doesn't matter what continent you're on. Aedes vexans is globally throughout. Very common mosquito across the board. Right on, Steve. I saw your hand raised. Right on, man. I appreciate it. So Aedes nipponi is very similar in morphology to Aedes vexans. I got some question marks here. Is it a variant or is it a species? Harbach in 2023 removed nipponi from subspecies status to species status, introduced into North America maybe around the time of Y2K. It has similar habitats as we know Aedes vexans to have. Sunlit habitats, flood pools, wooded floodplains, ditches, swamps, and the party goes on and on and on to dredge pool sites, bogs, and woodlands pools. So let's talk about dispersal strategies. We're at fault again. Here we are, anthropogenic. It's our fault. This mosquito is moving around the globe based on our behavior and the things that we need and want. It's thought of distribution currently. Its native range is basically the Korean Peninsula and maybe some areas in China. The thought of distribution of this particular mosquito, Aedes nipponi, there are some instances in Canada, in the U.S., like in Iowa, the possibility we're seeing character states in New Orleans, even in Chatham County, Georgia. We're seeing some similarities in Denver, Colorado, in that area, and then even in like Pasco County, Florida, in the upper central counties of the peninsular area. So we're seeing a wide range of possible distribution of Aedes nipponi. This is an ongoing project with the USDA, multiple different groups from the University of Seoul, some folks in Ontario, some individuals in Europe and Italy. And so we're kind of tying the knots on all of this work, trying to figure out who's who and what's what. But the pathogen transmission profile of this mosquito is Japanese encephalitis virus, which is pretty interesting. When we're thinking about JEV and its possible encroachment within pigs in the United States is a big, huge thing that the USDA and the CDC are constantly looking at across the board. So this mosquito is actually kind of important for us to shine the light on and figure out what's actually happening with this mosquito. So Aedes nipponi. So when we look at this mosquito initially, I'm looking at it, I'm like, man, this is Aedes vexans. It's a medium to large species. Its proboscis is brown with white scales all the way throughout. We've got banded tarsi, basal white bands, and then we've got bands on the abdomen. And that's about the where it starts to get interesting. We're starting to see these phenotypic expressions and scaling patterns in vexans populations here in the United States, where when you look at number 4 with those red arrows, and let me see if I can get my little cursor here. Oh, right here. Right down through here, you see this line going all the way down? And these are photos that were given to us of an Aedes nipponi from the Korean Peninsula by a postdoc there. So this medial line, if you're seeing this, we would really like to hear your story. It doesn't matter what continent you're on. We would like to talk to you about this mosquito, Aedes vexans, what might be a variation of Vexans or what might actually be the species Aedes nipponi. So these abdominal turgets banded with slight medial stripe of pale white scales going all the way down. And like I was saying, that there is ongoing work. So this is real time. So there's actually some projects that we kind of talked about today that are ongoing, like the Anopheles stevensii or the Aedes coriacus, the Aedes togoi, and now Aedes nipponi. These are things we're currently working on to find out what's actually happening so we can disseminate this information to everyone as a group so the information is available. And this is about to wrap it up. I can't believe I almost stayed on time, which is quite abnormal for me in some instances, especially with this topic. Um, I would like to thank the Southern Integrated Pest Management Center and the USDA for their continued support of beacons, and also for Gulf South Vector for supporting us as a group and including us in disseminating information to groups such as yourselves. And we'd also like to thank the Florida Medical Entomology Lab for supporting our website and dashboard that we introduced you to, and the long list of contributing agencies. We could not have done any of this important work without the support of our stakeholders and partners, and enthusiasts. There's a wide range of individuals out there that supply us with specimens and just help us kind of move this storyline along. My information is on the slide here in the at least my bottom right hand corner. My name is Mike Riles. That's my direct phone number. You can call me any point in time. If you're overseas, just put a 1 in front of it. And that's my email. You can contact me there all day long, mriles@central.com. I'm super excited about the distribution and migration of mosquitoes in the Western Hemisphere. If you're excited like I am, please reach out to me. Let's start a conversation across the board about what makes us excited about public health and mosquito species. I appreciate everyone's attention across the board. It looks like we got like 140 people up in here. That's awesome. And I want to throw a direct shout out to Claudia. I appreciate you inviting me to come in and share my passion with the group. Thank you so much.
We are so grateful. That was an amazing, presentation, and I absolutely love your enthusiasm. Right, you can tell when someone loves what they're doing. It's so fabulous. And really thank you for everybody for attending. And we do have some questions in there, so I'm going to kick it over to Astasia, so she can go through these questions. So I want to make sure I don't miss any, and she's good at this.
Okay, the first question, Mike, you mentioned the mosquito key books behind you, and someone asked, where can they get those?
Oh, so you can pick the new national key, which just came out. You can pick this up on UF Press. So the University of Florida Press, you can go online, and if you just put the North American Mosquito Guide and do UF Press, it should pop up in your Google search. It runs about $90.
I think we've got some more questions on there too.
Okay.
Stacey, you're on mute, just in case.
Sorry, Claudia.
It's okay.
Is the Atomia squabipens present in South America? And if yes, where can they check for it?
Yes, that mosquito is in South America. Let me run through this. I probably should have just popped out of it, maybe. Oh, I went too far. There it is. Yeah, so this particular mosquito is associated with water lettuce. So anywhere that you would find water lettuce, you will probably be a really good indicator of the presence of Aedeomyia squamipennis. One thing that I will make note of is the sunlit rays in swamps. they seem to congregate around areas that have this type of vegetation. So if you have like fallen logs and trees and things in the swamp and you've got your muck boots on and you're out there looking for things, look for that ray of light hitting up against those types of areas where the water lettuce or maybe duckweed or something like that is prevalent. And where that sunlight's hitting warms that area up. I've had super success about finding the larvae in spots like that. I hope that helps.
Okay. Hi, Mike. I'm with the San Francisco Health Department out in California. Thank you for an excellent presentation and putting so many new to me invasives on my radar. As someone in a region of California without much information exposure to these species, other than Aedes aegypti and Albopictus, are there any mosquitoes from this list that you feel are most susceptible to naturalization into Northern or Central California?
Yeah, most definitely. So here is the mosquito Aedes nodoscriptus, and you're going to find this mosquito in containers um, in small populations, like in San Diego and north. She has been found, and then so this is also a container inhabiting mosquito. You can also get in touch with your ports of entry and do some really interesting work partnering up with those larger municipal groups. Sometimes they're more economical driven. They could be ran from a federal standpoint. Um, Contacting those folks and getting some surveillance going on to put up some sort of surveillance wall to mitigate something that might come in. So when you're thinking about container inhabiting mosquitoes, you can have things like Aedes vegetatis come into that area. It could be a wide range of different species that could pop up, something that we might not even expect. So like that range of surveillance that we can set up in our ports, or at least the conversations that we could have with folks that manage our ports of entry, would be extremely an amazing step in your surveillance programs to take a look at. Currently, the invasives that I know of in California up to Washington are the Aedes nodoscriptus, the concurrent Aedes aegypti issues you're having there, and then you have Aedes togoi. But her habitats are so interesting, where they're like upper shelf saltwater tidal pools that mix with rainwater coming off of elevations. I don't really see her expansion getting too crazy, but you should always be ready for something that might happen, and setting up your correct surveillance strategies to reflect the ins and outs of commerce in your ports of entry would be awesome.
Okay. Someone asked, why would they have an issue with having no mosquitoes in some of their traps? Primarily their CDC light traps and their gravid traps.
Um, Let me see that question again. Can you?
So she said, why do I have no mosquitoes in some of my traps? CDC light and gravid traps.
Oh, okay, okay. So it could be a wide range of things. It could be a seasonal push. It could be they haven't been in the round. It could be wind pressure. There could be a several abiotic factors that can impact a trap not having mosquitoes. My question to you would be, what was actually in the trap? Was there anything in the trap? What were there any moths in the trap? Did you have any bycatch? Was the fan oriented in the correct way? Sometimes you can depolarize your batteries, and then your fan could be going the wrong way, which I've seen in some instances. And so there could be a number of different factors. If you want to contact me, we can talk a little bit deeper about your data and how you have your things set up, and we can talk about that all day.
Have you seen any interbreeding between invasive and native species?
Uh, I mean, so there is I mean, I think there are the instances of frolicking amongst species, right? If there's some hybridization going on, that would be interesting. Most of the time, those hybrids aren't very prevalent for oviposition, and they're fairly sterile. I think in the instance of that question, framing it would be Aedes aegypti and Aedes albopictus. I briefly mentioned the satirization impacts that ecological adaptation was going on, where Aedes albopictus actually, through its own natural sterile insect technique, pretty much pushed these mosquitoes back. Now, what was there an inbreeding and the possibility of a hybrid being pushed into the environment? Most likely not. A lot of times when you have hybridization like that, it doesn't really stand the test of time, and it genetically drops off.
You mentioned blood-fed Culex ink and Culex lac are of interest if we collect. Are any other adult specimens beacons? Are there any other adult specimens that Beacon is looking for? And if we collect fed or adult specimens, is there any specific handling that you suggest?
Oh yeah, so that's a very wide range topic, and I suggest whomever wants the correct answer for that. It could be fairly long-winded. We do have some standard operating procedures. I suggest that person who asked that question to reach out to us as a group, and we can tie in all the loose ends and get them squared away.
Okay. What harm does Aedes pertinax cause or is likely to cause in Florida that makes it invasive versus just non-native?
Yeah, so that's a great question. And, when we started this, we defined what was invasive versus native. And on our slides, we have like what its native range and what its invasive range would be. To be honest, this mosquito, its transmission is fairly ambiguous. We don't know its host biting preference. And even though its collections are apparent and in high abundance, we just don't have a lot of information about its status, whether it would be invasive and cause harm to us from either a public perspective or from an economic pressure or something like that. So that's a great question to redefine what we're actually talking about versus native versus a non-native versus an invasive.
Okay. I have a follow-up to the California question. We currently have passive surveillance for Aedes aegypti in San Francisco. What about Aedes aegypti given its suitability for cooler climates?
What was the other species?
Aedes japonicus? Sorry,
Japonicus.
japonicus. Thank you.
Right. So, um, coastal areas for Aedes japonicus, her numbers have always been low. She's generally a temperate mosquito, but we're seeing her adaptability to subtropical climates. Um, when you think about this mosquito, it's usually in more kind of wet type habitats. So if we would see this mosquito, you would see it in Northern California, Oregon, like Washington State, like things below the San Francisco line, like you're going down all the way to Bakersfield, crossing over to Orange County, something like that. You might see little small pockets if that mosquito ever got introduced there. There are some interesting papers that are available on her ecology and her adaptability. If you shoot me an email, I'll slide those articles to you, and we can chat about what her parameters are.
What kinds of considerations to have when trying to ID Culex that are blood fed, with how that changes the color of the thorax and abdomen and the shape of the abdomen banding?
Yeah, that's a great question. And when you're doing arborvirus work, you're using very specific traps, right? So and unless you're using like wide bore aspiration, you're using very specific traps that catch very specific mosquitoes. So you're really going to narrow down what's actually there, right? If you're pulling mosquitoes in host seeking traps, that's a needle in a haystack. If you're initially taking pulls from host seeking traps, um, that's that's kind of tough. I would suggest, some sort of field aspiration in the sites that you think that these mosquitoes might be at. You can target specific habitats. That really tightens the noose on blood-fed mosquitoes. Yeah, and then you're grabbing more of what you actually might want. And then gravid traps are very specific for like pipiens and Culex tarsalis, or your Culex coronators and your Culex salinarius, some of the things that we would like to be pulling for something like West Nile virus. Now, if you're pulling mosquitoes for things like dengue and chikungunya and things like that, then again, you're using very specific trap mechanisms to collect those mosquitoes, and you can use the mechanism to decide, hey, I know I'm at least going to be having this. So when looking at extended abdomens that are blood fed, you really want to remove yourself from any abdominal characters. You want to dial in on like Culex if the acrostitial setae are present. You want to look at the instance of banded legs or banded proboscis. You really want to take a look at those thoracic areas. Are you seeing those intergeminal spots that we spoke about? Are there scales present? In some instances, when those abdomens are elongated, those bands tend to disappear, and it can be seriously tough.
Do you think the limiting factor currently for range expansion for invasive Aedes is human-mediated transport or climate habitat suitability?
Both, both
As a,
in combination. So we're going to be moving stuff, and we're going to be moving stuff all the time. And not only that, we're hoarders as a society, and those things get moved. When we move from one home to the next, we've got 4 or 5 storage units. Then we're moving all of that stuff, and then you're thinking about all the commercial properties and all the movement from commerce, from tire trades to phyto type nurseries and things like that are shifting plants here and there. Um, uh, and as the climate kind of changes for us, the cycle of the earth, things are starting to warm up, and things are not as cool as they were in some instances. That's going to help the establishment and the adaptability to new temperate zones as things start to get warm.
okay. Those are other questions I see in the
Cool.
chat right now. Claudia, back to you.
We're super, yes, thank you again. I was going to show everyone our next series. Let me pull it up real quick if you're still online.
Do you want me to
We have
stop
some,
sharing? Do I need to stop
yes,
sharing?
thank you so much. Yep, if you could stop
Yep.
sharing real quick,
Here
I'll
we
go
go.
ahead and put it up so you guys can, there's a QR code there for you. We actually had our first webinar just this week, but we have another one coming up next week. And so we're talking about emergency preparedness. And so mosquito abatement is a huge component of mosquito abatement, as well as rodents and pet sheltering and all the different things that are related, or at least vector control can be related to. So you can also go to vectoroutreach.org, as I had mentioned, and go under events, and that'll be there. But if I'm keeping it up, you can use the QR code with your phone and register for it. It is free. I think it's going to be excellent. And you can see we're also including some case studies. And in fact, today I just finalized, so Puerto Rico, that unit is going to talk about their dengue epidemic from a couple of years ago. So that'll be on July 15th. It looks like we have a question, hands up is out there. If you can put that in the chat, that'd be great. And then we're gonna go ahead and wrap it up, and let's see if the question goes in. Otherwise, I did put Mike's email in the chat and you can email him directly. So with everybody, thank you so much for spending 2 hours and 20 minutes with us. And Mike, thanks again for all your knowledge that you're sharing with everyone.
Hey, everybody, have a great weekend. Thank you so much.
Thank
Much love.
you. Thanks so much. Bye.
Bye.